Hello everyone, my name is Shreya Kumarisha and today I'll be presenting a research paper based on microglia modulate amyloid beta dependent astrocyte reactivity. So microglia is actually the immune cells of the brain and astrocytes are the type of support cells. Amyloid beta is a type of protein which form plaques outside neurons and tau forms tangles inside neurons. Now Alzheimer's disease is usually explained using two main proteins, the amyloid beta and the other one is tau. So, for decades, scientists blamed Alzheimer's for a single culprit called amyloid beta. Now, billions of dollars went into clearing amyloid beta plaques from the brain, and yet almost every drug failed. And then arises the question, what if we were fighting the wrong battle? So, this paper shows amyloid beta alone is not enough to cause harmful astrocyte reaction. Microglia is the one which acts as the deciding factor. So now let's look at who is actually driving the process in this paper. First is amyloid. First, we have the microglia. In this study, they behave like the master switch. They are activated especially through the receptor TREM2. Here, the entire pathological cascade begins. Next is amyloid beta, which this paper highlights. The thing which this paper highlights is that it is the only initial spark. On its own, it actually cannot push astrocytes into a reactive or a harmful state. Then comes the astrocyte reactivity. When the signal astrocytes enter into a reactive high GFAP state, this reaction mode, astrocytes become less supportive and they actually start contributing to neurotoxicity. So they act as the main effector of the damage. Now, finally, this chain leads to tau pathology. This is a state that directly correlates it into neuron loss. Now, under the microscope, you can see the transformation. On the left, a resting microglia bushy with branches is there, and on the right, the same cell activated. Now it has become compact and rounded. Now, inside panel B, a key protein called NF kappa B has moved into the neurons. That means inflammatory genes are turning on. And you can see astrocytes nearly in green. The activated microglia releases cytokines that signals these astrocytes to become toxic. Now, earlier Alzheimer's was seen mainly as a neuron based disease. The idea was that amyloid plaques directly cause tau tangles and dementia. But it could not explain why patients with similar plaques levels show very different symptoms. So, clearly, something else was happening here. So, this study shows the missing link between microglia turning amyloid beta into harmful astrocyte reactivity. Now, here they used a multi-model design, a study design. It actually means instead of relying on just one type of measurement, the researchers combined PET imaging, CSF markers, and blood biomarkers. So, in this study, the authors used two complementary cohorts. One is the cohort A, which included 101 participants who underwent PET imaging. They measured amyloid plaques, tau tangles, and also microglial activation. This allowed them to visualize the pathologies directly living in brains. Now, cohort B focused on fluid biomarkers. So these two together, they gave a full picture from cells to the system. Now, the key finding of the paper is what they call the switch mechanism. This study shows that amyloid beta by itself does nothing. It cannot trigger astrocyte reactivity. But when microglia are activated, especially when TSPO and TREM2 levels are high, the entire relationship changes. Suddenly, the same amount of amyloid now produce strong and clearly reactive astrocytes. So, microglia basically acts as the on and off switch. Without them, amyloid is absent, and with them, amyloid becomes harmful. Also, in this interaction was statistically very strong with a p-value of 2 into 10 to the power minus 4, which is very good. Now, this paper describes this as a stepwise pathological cascade. First is the trigger step where amyloid beta starts accumulating, but on its own it stays largely silent. And step 2 is the activation, third is the reaction, and the other is the damage step. Now, impact of microglia on reactivity. When two individuals have the same amount of amyloid beta, their astrocyte reactivity is completely different depending on the microglial activation. So here it shows low microglial activation has lower impact and the higher activation with higher reactivity. So this study makes it clear that clearing amyloid alone won't work. The real target is the interaction between the microglia and astrocytes. So future treatment should focus on turning microglia pathways like TREM2 and blocking the inflammatory signals that push astrocytes into a toxic state. Thank you.